Yeah, today I'm going to be talking about how we are using high-throughput phenotyping at SRA to address some of the long-standing industry issues. This research is fairly new. We started this about 2 years ago, but it is based on a few years of physiological research in sugarcane. So it builds on all those physiological knowledge. So our main purpose of this research is to translate that vast knowledge in sugarcane physiology to actual practical outcomes in the breeding program. So just to give a snapshot of the Australian sugar industry, it's not as big as the GRDC, but there's about 36.5 million tons of sugarcane that's been harvested every year and it's grown across 370,000 hectares of land. It's the third largest supplier of sugar globally, and with an estimated annual value of more than $1.5 billion. SRA is the principal provider of R&D for the Australian sugar industry, with the major goals of driving profitability and improving sustainability of the sugar industry. So the main focus of phenomic research at SRA is to try and improve the rate of genetic gain. So if you look at the rate of genetic gain for TCH, for example, that is tons of cane harvested per hectare, which is basically cane yield. It's been fairly stagnant for the last few years, and if you look at for CCS, which is the actual amount of sugar that's been extracted from sugarcane, the trend for rate of genetic gain is actually negative. So the major focus of research at SRA at the moment is to try and reverse this rates of genetic gain trends through various methods. One of them is modernizing the plant breeding program. So plant phenotyping and high-throughput phenotyping is one of the integral components of this modernization process. So in some ways, that the phenotyping is going to bring about some changes to the breeding program is by improving the precision of phenotyping, which should aid in making better varietal selection decisions. And in phenotyping, important traits of interest, for example, NUE and water use efficiency, which normally would be very difficult to phenotype in a breeding program using traditional methods. And then to improve our understanding of sugarcane plant physiology so that we could target specific traits for specific growing conditions. And the hope is that the phenomic research will enable high-resolution linkage mapping and GWAS studies for sugarcane improvement. So as I mentioned, it's been going on for 2 years, this research, but we have managed to set up a fairly good aerial phenotyping platform. We have 5 aircrafts of various sizes. We have a number of qualified pilots. We have a range of sensors from multispectral to thermal to hyperspectral cameras. We do about 100 flights a year, cover about 20 plantings a year, and generating enormous amounts of data. And all of this data is processed in a semi-autonomous workflow. And more importantly, all of this data is available almost instantaneously to researchers and plant breeders so that they could review and make decisions. So this is a typical workflow. I'm sure many of you are familiar with. So mission planning, image collection, mosaicing, point cloud and DSM construction, then extracting plot level data, and a lot of ground truthing as well. But more importantly, all this data is available online across the organization to researchers and plant breeders so that they could make decisions as the plant grows, rather than wait until the end of the crop cycle, which is 12 months for sugarcane. So very much our focus at the moment is to try and improve the early stage selection of the SRA breeding program. So this is a general overview of the different stages of the SRA breeding program. In stage 1, we have got about 225,000 seedlings from a few different crosses, which are assessed in single row plots for cane yield, cane fiber, and cane sugar. And from stage 2, around 2,500 clones are advanced to stage 2, which are then assessed in single row 6-meter plots for TCH, cane sugar, and some appearance grades and diseases. From stage 2, about 150 clones are advanced to stage 3, which are then grown, assessed in, and grown in 4-row 10-meter plots. So the stage 3 assessment is actually more aligned with commercial pure stand conditions that you would normally see. So the problem with the stage 1 and stage 2 of variety assessment is that it suffers from low heritability of cane yield, and there are various reasons for that. One of them is it could be due to large measurement error that could arise due to a significant lodging event. Cyclones are pretty common in sugarcane growing regions. And the trials are very huge. It's about 3,000 clones that are of 2 replicates that are grown in around 7 to 8 hectares of land. So there's bound to be a number of unknown spatial effects. And another aspect is that clonal performance in single row plots can be highly influenced by competition effects. So what that means is clonal behavior in single row plots won't necessarily translate to multi-row plots or to pure stands. So it results in a poor genetic correlation between single row and multi-row cane yield. And this actually represents a significant wastage of resources and time for the breeding program. So we are using phenomics approach to try and address this issue. So we are trying to improve the precision of phenotyping. Currently, our focus is on the stage 2 of the breeding program, clonal assessment trials. And we are trying to move the selection earlier in the crop cycle so that before a significant lodging event or before the onset of significant competition effects. So our approach is to select for plant vigor and canopy temperature, indirect traits, indirect yield-related traits, and use that in a multiple trait selection index for variety selection. So with each of these objectives, we are trying to address at least one component of the genetic gain equation. So with, for example, with points 1 and 2, we are trying to improve the heritability, therefore improving the selection accuracy. With points 3 and 4, we are trying to assess novel genetic variation, therefore improving the genetic variance in the genetic gain equation. And because aerial phenotyping is non-destructive, and because sugarcane is a vegetatively propagated crop, there are significant opportunities to reduce the years per cycle in the breeding program. So with each of these objectives, we are trying to improve enhance the genetic gain of the breeding program. So in terms of methodology, we use RGB multispectral and thermal imagery to measure a few different traits, including ground cover, plant height, a number of vegetation indices, canopy temperature, plot gaps, and lodging. So we do this over the course of the crop cycle. Sugarcane is a 12-month crop. So for canopy temperature, we have to do that for multiple times a day. So all of this is to investigate what would be the best time to capture these traits so that we could maximize the genetic variation that can be captured. So in terms of validation, this was done last year. We got pretty good squares for manual plant count and aerial ground cover, manual plant height and manual aerial plant height, and for NDVI as well. If you look at the genetic correlations between ground cover and plant count, it's 0.84, and for plant height and DSM-based plant height, it's about 0.75. So we are pretty happy with the validations. In terms of, if you look at the heritabilities of some of the aerially measured traits, such as NDVI, DSM, ground cover, and are actually better than those that were measured manually. So we applied 3 different selection indices to this data. So the first selection was based on conventional selection that's currently being applied in SRA. And the second selection was based on an indirect trait-based selection. The third selection was a combination of selections 1 and 2. So when we compare the clonal rankings for selection 1 and 2, the R-square was about 0.4. And the clonal ranking for the correlation between clonal ranking for selection 1 and 3 was 0.76. So there's significant differences in the ranking of clones when we apply the 3 different selection indices. So if you look at the top 250 clones that would normally be selected from the selection 1, and compare it with selection 2 and 3, there were about 108 clones that were common between selection 1 and selection 2, and there were about 175 clones that were common between selection 1 and selection 3. So very much our next stage of this research is to try and test if we were to advance the uncommon clones from the selection 2, would the genetic gain be much better than the conventional selection method? Yeah, so for the next stage of validation, essentially we are trying to assess early vigor and plant vigor traits and assessing the translation from single row to multi-row plots and comparing it with the translation of TCH with single row and multi-row plots. So if it turns out the translation of early vigor traits was much better than the translation of TCH, then we could make recommendations to the breeding program to use a trait-based selection that should capture the clonal performance in the single row plots much better than the conventional method. So this is a related topic where we are screening for some NEV-related traits in the field. So this is a trial where we have 40 different genotypes and 4 different nitrogen treatments. One of them is a controlled release treatment. So we can monitor how each of these treatments develop over time. And we are actually getting pretty good correlations with developmental and growth-related traits and with NEV-related traits such as leaf nitrogen. We are able to pick up the strong genetic variance between the clones with a high degree of confidence. So moving forward at SRA, one of the objectives is to extend the precision phenotyping to the first stage of the selection breeding program, where we can monitor single seedlings in the field. So this will enormously improve the selection intensity and improve and consequently increase the genetic gain. And another related work that's been going on is providing some variety descriptors. For example, in this graph, we are describing potential candidates for release against known standards, for example, for rate of canopy closure, or weed competitiveness, or flowering and rodging propensity. So this type of description of candidate release cultivars would help breeders and growers to make better varietal selections. And just some work that we are starting to embark on is assessing some canopy spectral signatures for sugars and chlorophyll content. So this is our team. Prakash is in the audience, Jaya and Xiaoming and Jason. And thankful for the technicians, Johan and Bill. Yep. So thanks for your attention and happy to take any questions.